All right. Hi, everyone. My name is Brianna Sear. I'm a graduate student of Juan Pablo de Rivera Vicari. And today my talk is going to be about the role of the inflammasome in sexual dimorphism and aging. All right, to begin, my project focuses on inflammaging. And inflammaging is this low level of chronic inflammation that occurs due to old age. So as we can see here, as we age, we experience, fluctuations of our inflammation as maybe we get infected with a disease or even stress. So this can all slowly start to build up over time and leads to a heightened kind of baseline level of inflammation as we enter old age. And this is an issue because this heightened inflammatory environment can lead to changes in cellular function. So here, this is mainly just showing, central nervous system cells. So we can see as we increase with as we age, the more pro-inflammatory cytokines are also increased. And as we age, the more pro-inflammatory cytokines are decreased. And so this has a variety of effects on a variety of cells. Oligodendrocytes, there can be defects in remyelination, endothelial cells, neurons, astrocytes, microglia, and the choroid plexus. So all of this will lead to a sort of dysfunctional environment. So what is actually producing these various factors? What is creating this inflammatory environment? And of course, the answer is a wide variety of factors. As you can see here, things such as changes in the gut microbiome, infections, just intrinsic immune cell defects can all lead to a number of different dysfunctions, including mitochondrial dysfunction, epigenetic alterations, increased oxidative stress, and this in turn will lead to the release of these pro-inflammatory cytokines, these inflammatory factors. What my project is focusing on specifically, the inflammasome activation, and all of these things can lead to inflammaging. So why do we care about investigating inflammaging? I know I mentioned that, it leads to a kind of dysfunctional environment. All of your cells start to become dystrophic, but what does this actually lead to? What are the actual consequences? And again, a wide variety of age-related diseases. So inflammaging does affect the whole body. So you can experience things such as cardiovascular diseases, diabetes, kidney disease, cancer, and of course, in line with my project, neurodegenerative diseases and cognitive decline such as dementia. Okay, so what specifically is the inflammasome? And the inflammasome is a multi-protein complex that is activated upon receiving one of those inflammatory signals. And it does in turn output another inflammatory signal. But first, to break it down, the inflammasome consists first of a sensor protein, which you can see here in the NOD-like receptor proteins NLRP1, 2, 3, and then there's also this absent in melanoma 2 sensor protein. There's also an adapter protein, ASC, and then the effector caspase 1, which when cleaved, it will then cleave the pro-inflammatory cytokines IL-1 beta and IL-18. Also, as you can see here, different inflammasomes are found in different cell types, but this doesn't mean that you can only find NLRP3 inflammasomes in microglia. You can find them in other cells as well. These are just the predominant inflammasome types found in each of these cells. So how does the inflammasome become activated? And it will receive, as I mentioned, an inflammatory signal. This can be something as a PAMP or a DAMP, which is a pathogen-associated molecular pattern, or a damage-associated molecular pattern. And so this is specifically also looking at the NLRP3 inflammasome. So you have this first kind of signal. It can also be an increase of extracellular ATP. And this is the first signal. This kind of leads to the expression of the inflammasome proteins. And then it needs a second signal, such as lysosomal rupture, mitochondrial dysfunction, or reactive oxygen species. And that allows the inflammasome to oligomerize into its whole multi-protein complex. Once activated, the pro-caspase-1 cleaves, and once it is active, it in turn cleaves the proforms of the cytokines IL-1 beta and IL-18, as I mentioned prior. It also cleaves this protein here, gasdermin D, and the N-terminal of the gasdermin D protein actually gets inserted into the cellular membrane, and this forms a pore through which IL-1 beta and IL-18 can escape, and then go on to affect and signal to adjacent cells. Once there has been sufficient inflammasome activation, it can also ultimately lead to a form of cell death known as pyroptosis. And this is a very messy form of cell death. It's kind of almost like an explosion. It pretty much just dumps everything that was inside of the cell outside. It doesn't package everything up nice and neatly. Everything just kind of gets thrown out there. So this is known as the canonical pathway of inflammasome activation, but there have also been two non-canonical inflammasomes identified. This figure here only shows the caspase 4,5,11 non-canonical inflammasome, but caspase 8 has also been shown to be a non-canonical inflammasome. And what makes these different is that they do not form multi-protein complexes. It is just the caspase. It receives the signal, becomes cleaved, and then here, as we can see specifically, caspase 4,5,11 can cleave gasdermin D. Okay, so when the inflammasome oligomerizes into its whole multi-protein complex, it does so through ASC oligomerization, and that happens in a prion-like mechanism, so kind of happens very rapidly all at once. This then can be expelled into the, and this is actually known as an ASC spec once it forms the oligomer. You can see here the congregation of the ASC polymerization. So this can then be expelled into the extracellular space once the cell undergoes pyroptosis. And this is an issue because once out in the extracellular space, it can actually still affect IL-1 beta and IL-18 and can lead to further inflammation even after cell death. So a paper previously published by the lab has shown that inflammasome activation does increase with age. Here we received fibroblasts from a human donor, a healthy donor, at three different time points, age 49, 52, and 64. And a Western blot analysis was performed for the inflammasome proteins caspase-1 and ASC. And here, the lab was able to show that over time, as this donor aged, there was an increase in expression of caspase-1 and ASC. So suggesting that, yes, inflammasome activation does increase with age. So one aspect of research that is often neglected is actually the sexual dimorphism aspect of the immune system. It has been shown time and time again that there is differences in both innate and adaptive immunity between males and females. So, and this is also ultimately affected by aging as well. So in this figure, we can kind of see that as we age, there are different time points at which, so here in utero, there's increased inflammatory responses in males. And then inflammation is up in males during childhood, But then once we get to adulthood, inflammation is up in females, and then inflammation is up again in males in old age. So not only are there differences in the immune responses between males and females, but then that itself is also affected by age. So that brings me to my first big question, which is whether there is a difference in inflammasome protein levels between young and aged male and female mice. So my experimental approach to answer this question is fairly simple. I took young mice that were 3 months old and compared them to aged mice at 18 months old. So I removed their brain and turned it into a lysate to perform Western blot analyses. First, here I have the comparison of young females to aged females. And so I performed the immunoblots for 3 different inflammasome proteins, ASC, caspase-1, and IL-1 beta. And then these were all normalized to beta-actin. And as we can see here, for all 3 proteins, there is a significant increase in the expression of the inflammasome proteins from young to aged females. So this shows us that the inflammasome is activated in aging in female mice. Then I compared the young male mice to the aged male mice for the same 3 inflammasome proteins, and again, saw the same effect. There is a significant increase in the expression of inflammasome protein levels from when comparing the young to the aged mice. So again, this is showing us that the inflammasome is activated in aging in male mice as well. So then I wanted to try to directly compare the levels of ASC specs. And so I performed this protocol where I was able to perform a partial pyroptosome purification. So essentially taking the lysate and just isolating out all of the proteins that are associated with pyroptosis, the inflammasome. And then I was able to perform an immunoblot against ASC to see I guess the different types of ASC. So down here we have the monomers, the dimers, and the oligomers, so the specs. And once I quantified them, I did see that there, for both males and females, there's a significant increase in the levels of ASC specs during aging. Next, I went on to compare the difference between males and females. And here I have the aged females compared to the aged males. Again, investigating the same 3 proteins, ASC, caspase-1, and IL-1 beta. And upon quantification, I found that there is a significant increase in expression, a significantly increased expression of these inflammasome proteins in the aged females compared to the aged males. So this data is suggesting that females have a higher level of inflammation due to the inflammasome in old age compared to males. Now I compared the young females to the young males to see if maybe there would be a difference also at the young time point, and I found the same thing. There was a significantly increased expression of the inflammasome proteins in the young females compared to the young males, suggesting that even at this young time point, females have a heightened level of inflammation due to the inflammasome compared to males. So then I wanted to determine if the rate of increased inflammasome activation was the same between males and females. So I calculated the percent change from the young time point to the aged time point in both the females and males. And how I did this is the equation is just right down here. So it's the aged minus the young divided by the young times 100. So this will kind of give you the percentage different, how much it actually increased in comparison to the young time point. So looking at the actual rate of increase of inflammation between males and females, there's actually no difference. So this is showing basically that though females have an increased amount of inflammation, they don't increase in inflammation more than the males. I think this next one will kind of show it a bit better. So this was a paper published back in 2011, and this was looking at different inflammatory proteins, but they measured the levels from a younger time point to a fairly aged time point, and this is in humans. And this actually shows what I saw in my mouse data. So here is the males and here's the females. So as we can see, even at this younger time point, the males have less inflammation than the females, but then they both increase over time during aging. But if you kind of try to compare the slope, it looks like the rate is about the same. So all in all, this is showing that males and females do have inflammaging at the same rates. They increase about the same in comparison over time, but females just start off with a higher level of inflammation. Yes. Oh, yeah. So the question was asking if I looked at the young females in overoptimized model as well. And I did not. These were just normally, they were just going through the normal estrus cycle. They were just normal young female mice. I did take them during the diestrus time point to kind of, I guess, minimize any effects that the estrus cycle might have had, since it would be at the point of time in which the hormones were the lowest. But I did not look at the overoptimized mice. So to wrap up this section of my talk, what I have found is that inflammasome proteins are increased with age in the cortex in both female and male mice. The inflammasome proteins are increased in the female mouse cortex compared to the males at both young and aged time points. And the rate of increase of the inflammasome proteins during inflammaging for male and female mice is not significantly different. So they're inflammaging at about the same rate. Oh, yes. Yeah, so the question was if I looked at any cognitive differences doing cognitive testing with the male and female mice to see if the inflammaging actually impact, I guess, the cognitive function of these mice. I did not, but in actually, I guess kind of this slide is showing that a paper published previously by the lab looked in rats. And here they did do a treatment. So I guess this is kind of looking at the effect of the treatment and not necessarily between the young and the aged models. But this is showing that by decreasing the levels of the inflammasome proteins using probenecid, it was able to improve the cognition of these mice. So I don't believe our lab has published looking at specifically the difference between the young and the age time points in the cognitive abilities. I think for my second aim, I might investigate that, but I am also planning on doing a treatment. So I guess to sum up, I do not know if there would be a significant difference in the cognitive abilities between the young and the aged mice at these time points. Right, that'll be something that I definitely keep in mind, and you're my committee chair, so I'm sure we can push for that experiment to happen. Yeah, I mean, I was surprised that I didn't find any difference just looking at the male and female data. It just hasn't been done in the inflammasome, really. It's just a yeah, I'll have to look into that further. There probably is, and I maybe just haven't seen it yet. Right. Okay, so I'll go back over this slide since it was kind of maybe not explained in the best way. So a paper published previously by the lab looked at a treatment with probenecid, which is an anti-inflammatory drug, and they looked at the cognitive ability of rats. And here, specifically, we probed the lab. I was not a part of this paper. This was before my time. The lab probed for caspase-1, pennexin-1, and P2X7R. So these are all kind of involved in the activation of the inflammasome. The caspase-1, if you remember, is the effector caspase. And when looking at the levels for the vehicle treated compared to the probenecid treated, probenecid was able to cause a significant decrease in the levels of each of these three proteins. And then upon examination with the Morris water maze, those that were given probenecid showed increased cognitive ability compared to the controls. They had a shorter mean path length compared to the vehicles treated. And these are in aged mice, if that wasn't clear. Okay, so the treatment that I will be looking at in my project is IC100. So IC100 is a humanized monoclonal antibody against ASC. It has a low immunogenicity of 9%, which means it has a low chance of your body kind of reacting badly to it and needing like higher doses along the way. It also has fast binding and slow dissociation in the nanomolar range. It's safely dosed up to 300 milligrams per kilogram in non-human primates and in rodents, and it is able to penetrate the central nervous system, as seen by this figure here. So this, the way that we determined if it penetrates the CNS is by using radiant efficiency. So there were control mice and then IgG mice and IC100 mice. And so the IgG and the IC100 were tagged with a fluorochrome. And the way that radiant efficiency works is basically this fluorochrome will be like measured with a camera. And so here, looking at the brain, we can see that down here is the control. This is at 8 hours and 48 hours post-injection. The blue here is the IgG and the green here is the IC100. And so from this, we can see that IC100 does in fact have some penetration into the brain at about the same level that the IgG does. And this was also done with the spinal cord. And again, we can see that the IC100 penetrates into the spinal cord at about the same, I guess, efficacy as the IgG. Why would we want to target ASC specifically with IC100 when, you know, I did just show that probenecid looked like it had a pretty good effect? And the reason for this is that ASC is involved in every inflammasome complex. Well, I guess except the non-canonical inflammasomes, but we'll get to that in the next part of my talk. In addition to this, so we can target every single inflammasome complex with just this one drug, but Also, it can prevent the continued inflammatory activity of the ASC specs when they're expelled into the extracellular space after cell death. So even if you had like a really bad inflammatory reaction, a bunch of your cells pyroptosed, you could take IC100 and then you could prevent all of the ASC specs that are out in the extracellular space from continuing that inflammatory reaction. You can resolve your inflammation and be healthy again. Okay, so that leads me to my next question, which is whether treatment with IC100 can decrease inflammasome-mediated inflammaging. To answer this question, my experimental approach was to use just regular 3-month-old mice, 18-month-old mice that were injected with saline, the vehicle, and 18-month-old mice that were injected with the treatment IC100. Then after 3 days of the injections, the brain was removed, and then it was either used to become a lysate to do Western blot analysis or to be sectioned for immunohistochemistry. First, looking at whether IC100 is in fact effective in decreasing inflammasome activation and inflammaging, I examined 3 different inflammasome proteins. Here I looked at NLRP1, one of the sensor proteins, caspase-1, and ASC. And again, these were all normalized to beta-actin. Here we can see that in all 3 proteins, NLRP1, caspase-1, and ASC, we again see that there is that significant increase of the protein expression levels from young to aged. And then we can see that between the saline treated and the IC100 treated, there is a significant decrease. So this is showing that IC100 is effective in decreasing inflammasome activation during inflammaging. This, so it's 1 injection of IC100, and then 3 days later, the brain is removed. This is actually only males. Okay, so this last figure here, I again did an isolation of ASC specs. However, this only had an N of 1, so it is not quantified. This is just kind of for visualizing purposes. But here we can kind of see that within the oligomers, it appears that there is an increase in ASC specs from the young to the aged, and then it appears as though IC100 is able to decrease this. Of course, we need to increase this N to be able to quantify and to, soundly determine that IC100 is effective in decreasing the ASC specs. So now that we know that IC100 is effective in decreasing inflammasome activation, we want to see that it does also decrease IL-1 beta, because in essence, IL-1 beta is what goes on to affect the other cells surrounding and perpetuate the inflammatory signal. So looking at the young compared to the age, the saline injected, again, we do see that there is that significant increase from young to aged, and that when treated with IC100, it is able to decrease the levels of IL-1 beta. So it appears that IC100 is fairly effective at decreasing the inflammasome-mediated contribution to inflammaging. Yes. Yes. Mhm. Yeah, so the question was basically whether if I think IC100 could kind of reverse the cumulative effects of the inflammaging with just this one injection of IC100. I don't think so. I think once you've accumulated and gotten the damage, that damage is done. I don't think you can really reverse that. But what IC100 can do is prevent from I guess, continual increase. So if you think back to the graph that I showed at the beginning with inflammaging, how it kind of slowly increases over time, it can kind of stop, I guess, or try to prevent that increase in inflammaging. And maybe you've already increased from, like, if this was your baseline, maybe you've already increased to here. I mean, IC100, I don't think will make you go back down to here. You've probably gotten maybe some damage. But what it can do is keep it here as opposed to letting it come up to here next. I hope that answers the question. Okay, and then I thought I saw another hand. No, we're all good? Okay. Now I investigated the role of the non-canonical inflammasomes in inflammaging and whether they also contribute. I did do this in the treatment model. So looking at the 3-month, the saline injected and the IC100 injected aged mice, we can see that there is no difference in expression for caspase-11. The IC100 treatment didn't do anything, and it doesn't appear that caspase-11 really contributes to inflammaging. There's not a significant increase from the young mouse to the aged saline injected mouse. However, we did see something very interesting with caspase-8. So caspase-8 did have a significant increase in protein expression levels from the young to the aged mouse, suggesting that caspase-8 does play a role, does contribute to inflammaging. But IC100 was able to decrease the levels of caspase-8. And this was interesting to us because IC100 targets ASC, and these non-canonical inflammasomes are supposed to not form multi-protein complexes. So to investigate further, I performed an immunoprecipitation against ASC. And here I was actually able to see that it does appear that caspase-8 is interacting with the canonical NLRP1 inflammasome. So this was really interesting. No one's shown this effect before, that the non-canonical inflammasomes could interact. Maybe it's just an interaction, maybe it's involved in a whole multi-protein complex. We're not really sure. We just know that there is an interaction here. Lastly, for this section of the talk, I performed some immunohistochemistry to see if these inflammasome proteins were, in fact expressed in different cells of the central nervous system. So here I stained with nu-N to stain for neurons, and then I stained for the different inflammasome proteins that I've talked about, just discussed. So ASC, NLRP1, caspase-1, and caspase-8. So here we can see, so the inflammasome proteins are in red and the nu-N is in green. And we can see here that there does appear to be expression of the inflammasome proteins with neurons. And here, the expression here isn't as strong, but there is still some, caspase-1 we can see, and with the neurons, and same with caspase-8, just a little bit of staining. So to conclude for this section of my talk, I have found that IC100 reduces the levels of inflammasome proteins in the cortex of aged mice. I found this new interaction between the non-canonical caspase-8 inflammasome and the canonical NLRP1 inflammasome in mice, and the inflammasome proteins are present in neurons in the mouse cortex. I specifically wanted to examine the role of microglia in inflammaging and what role it plays. This is because microglia are known to change with age. So this is known as microglial priming. And once microglia become primed, they upregulate a lot of pro-inflammatory factors. They have defective phagocytosis, aberrant kind of cell communication. So I wanted to determine if maybe the inflammasome is involved in some of these changes. IL-1 beta was found to be upregulated in primed microglia, and ultimately changes in microglial activation has been linked to cognitive impairment. So maybe this is what we need to be targeting for inflammaging in order to prevent development of neurodegenerative disease. And lastly, it has been shown that microglia phagocytose ASC specs, so there is an interaction there with the inflammasome. This was some more staining that I did with IBO1. So because these are aged mice, I can't say for certain that they're microglia because macrophages have been known to infiltrate the aged brain. But here I can see that there is colocalization of these three inflammasome proteins, ASC, caspase-1, and caspase-8 with the IBO1 stain. I'm currently still working on imaging these sections, so I don't have the 3-month-old data here, but hopefully I will have that soon. But it's promising that I do see that at least in the aged cortex, it appears that this IBO1, these microglia and macrophages are colocalizing with the expression of these inflammasome proteins. So that brings me to my final question, which is, what is the contribution of ASC specs to inflammaging and microglia? So this is my overall hypothesis because this is really the meat of my thesis. I guess let me walk you through it. Basically, we start off with our young, healthy brain. You have, your microglia, your astrocytes, neurons, and you have your oligodendrocytes forming the myelin. However, when we age, there is the increased inflammasome activation. And this could occur in all of the cell types. All of them have been shown to have, an inflammasome. So during inflammaging, eventually these cells are expressing a heightened amount of inflammatory factors. If the inflammasome is activated enough, then the cell will undergo pyroptosis and expel ASC specs into the extracellular space. And then these ASC specs can in turn cause more activation, specifically to microglia, and cause inflammasome activation in the microglia, which will lead to pyroptosis. So my theory is that the microglia are kind of stuck in this vicious cycle of inflammasome activation and cell death. And hopefully IC100 can come in and block the ASC specs so we can break this cycle. To first answer this question, I needed to determine that ASC specs do in fact activate the inflammasome in microglia. So using an immortalized cell line, I plated them and applied ASC specs for 4 hours and then collected the media for lactate dehydrogenase or LDH measurement, and then also for caspase-1 activity. So LDH is released upon cell death. So this is kind of my measurement to determine if cells have died. And the caspase-1 activity is to determine if there has been activation of the inflammasome. Here actually showing the different concentration levels that I used. I started with 3 micrograms per milliliter up to 300 micrograms per milliliter. And here I found that at around 200 micrograms per milliliter is when we start to see significant activation of the inflammasome. And this is also mirrored in the LDH release. At about 200 micrograms per milliliter, we see a significant increase in LDH release and therefore cell death. Because LDH is just released by any kind of cell death, I can't say for certain that this is pyroptosis. It could also be apoptosis, but since it is kind of, mirrored with the caspase-1 release, I feel that once I investigate further to determine if, there's an increase of gasdermin D, which is what leads to pyroptosis, that I can say for certain that this is pyroptosis. However, I've yet to perform that experiment. But now that I know at what concentration the ASC specs have a significant effect on the microglia, I wanted to test whether, maybe what the ASC specs are doing. Are they, the first signal? Are they causing induction of the second signal? What exactly are they doing to the microglia to cause the inflammasome activation and cell death? So here, using the same microglia, I did a pretreatment with mitotempo, which is a mitochondrial superoxide quencher. And this pretreatment was for 1 hour. Then I applied the ASC specs at the 200 micrograms per milliliter concentration, since that was the lowest concentration at which I saw an effect. And then again, left them for 4 hours as I did previously, and then stained the live cells with mitosoc spread in order to stain for the mitochondrial reactive oxygen species. And then I saved the media to perform a caspase-1 activity assay to measure the inflammasome activity. First, looking at the caspase-1 activity, when I compared the controls to the ASC specs, there is still that significant increase. So again, validating my previous results that the concentration of 200 micrograms per milliliter does cause a significant effect on the inflammasome activity of these microglia. However, the specs with the pretreatment of the mitotempo were about the same as just the specs alone. So this kind of indicates that the mitotempo pretreatment did not do anything to prevent the activation of the inflammasome. And then here, looking at the actual mitochondrial ROS, comparing the control to the ASC specs, there is no difference, which means that these ASC specs are not inducing any sort of reactive oxygen species production within mitochondria. So all in all, these specs do not have an effect on mitochondrial reactive oxygen species production. And overall, for this section of my talk, I found that ASC specs do cause inflammasome activation and cell death in microglia at concentrations of 200 micrograms per milliliter and higher, and that the ASC specs do not cause an increase in the mitochondrial ROS. So this is what I am currently working on right now. So this is just the kind of preliminary data that I'm working with, but it's very promising so far, and I'm excited to see what I see with my future experiments. Next, I plan to apply the ASC specs to primary cultures to get, I guess, a more translational feel for ASC specs on microglia, and I also want to treat with IC100. So I'll be measuring the inflammasome proteins to determine if treatment with IC100 can affect the effects of the ASC specs to the microglia. I'll also be performing live imaging to kind of see how the microglia are interacting with the ASC specs. And then ultimately, I want to administer the ASC specs to mice and treat with IC100. So to do all of this in an in vivo model, and ultimately perform some cognitive testing. So hopefully to see if there is an effect with that, and also to characterize the microglia via flow cytometry to see how the microglia have reacted in an in vivo model. Okay, with that, that brings me to the end of my talk. I want to thank my lab, Dr. Keene, Pablo, my mentor, Roy, my lab mates, Nathan and Xavier, also the Dietrich Bramlett lab, Dr. Dietrich, Dr. Bramlett, and Nadine. She actually helps me a lot, my committee, and the neuroscience program. So I'm open to take any questions now. Thank you all for listening.
Okay, my people in Zoom, do we have any questions in Zoom? Do we have any questions here in the auditorium?
Hi, Bree. Nice talk.
Thank you.
How are you actually going to administer the ASC specs to the mice? Is that something that is tail vein injected? Are you doing stereotax directly into a specific brain region of interest?
Yeah, that's a good question. So the plan right now is to do, I believe it was an interarterial injection. One of my committee members, Dr. Raval, said that she does them with rats, and so she could teach me how to do it in mice. So that is the current thought. However, if that doesn't work out, then the other thought would be maybe to do tail vein, and then even possibly stereotax. But stereotax would be the last kind of idea, because we don't want to damage the brain, since we're kind of looking at the brain. And that would cause an inflammatory reaction. So it might kind of skew the data if we do it that way.
Very nice talk. Thank you very much.
Thank you.
I guess you're just specifically monitoring inflammasome signaling in the brain. And I noticed you also did spinal cord injury. In these animals, are you also taking tissue from other organ systems and looking at inflammasome signaling? Is this just an overall increase in inflammasome activation in the animal, or is it restricted to the nervous system?
Yeah, so I've only been looking at the brain, but there definitely is increased inflammasome activation in other organs. I believe the paper that I read was specifically looking at, I think it was like the pancreas, maybe. I don't remember for sure, but there have been other papers out there that have looked at the inflammasome in other organs during aging and have also seen an increase. I personally have not taken any other organs and examined those.
Yeah, it's an important question, since a lot of people have not looked, as you mentioned already, in terms of inflammasome signaling in aging. And so it'd be kind of interesting, since you've got the aged animals, in addition to the nervous tissue, you might want to take the other organ systems. Now, the mitochondrial reactive oxygen species, that's a good experiment. I just wonder if you should look at some other indicators of mitochondrial dysfunction. And we've certainly got some world experts at that look at mitochondria. So you might want to have some conversations with those individuals.
Yeah, I think that might be interesting. That might be a project for someone else in the lab, since I'll ultimately be doing in vivo work. I would be interested in investigating the mitochondrial more, because actually for the work that I did in undergraduate, I utilized a seahorse analyzer and measured cellular respiration in microglia. So I think it would be really interesting to see maybe if I see the same effect, because I was specifically looking at activated microglia and seeing that the rate of glycolysis and oxidative phosphorylation is different between like quiescent microglia and activated microglia. So it could be interesting to see if you see that same effect with primed microglia, if they become more reliant on glycolysis rather than oxidative phosphorylation in old age.
So
Yeah, that's fair. It might have an effect on the mitochondria. It could cause dysfunction in the mitochondria, but it doesn't seem, at least acutely, to affect the rate of mitochondrial reactive oxygen species production. That clears it up.
Thank you for the talk. So you showed that, even with a single dose of the IC100, that these different inflammasome pathway proteins are downregulated or, detected at lower levels. Is there a way to check whether pyroptosis is also decreased?
Yeah, I could do a Western blot for gasdermin D to see if the levels of those are increased. Gasdermin D can be a little tricky to stain for sometimes. I think I might have actually done it and maybe the results weren't significant. I can't remember. But that's one way that I could do it to determine the pyroptosis.
Or just like reduced cell death in the brain or something like that. It's like that.
Yeah.
Okay.
All right, I think we're all good. Thank you everyone for coming to my talk.